TIMER2 is a web platform to help cancer biologists investigate the association of their interested gene expression, mutation, somatic copy number alteration with tumor immune infiltration, and together how they influence on patient overall survival. TIMER2 could also help analyze non-immune-related associations, such as differential gene expression between tumor and normal samples, and association between specific gene expression and the patient clinical outcome, and etc. It can also help estimate the immune cell infiltration for user-provided expression profiles by 6 state-of-the-art algorithms. Next, I will introduce how to utilize TIMER2 in cancer research. For example, ARG1 is predominantly expressed on tumor-associated macrophage, which is a well-known immunosuppressive cell type. And we can use the gene module here to test whether the expression of ARG1 is associated with T-cell infiltration in tumors. So we can go to the gene module by click here and type the ARG1 gene. And we can select the immune cell type we are interested. Here I will use the CD8 T cell as an example. As you can see, TIMER2 output this heat map where the rows are the different cancer type in TCGA dataset, and the columns are CD8 T cells estimated by different algorithms, such as TIMER, EPIC, and etc. And the values in this heat map is the partial Spearman correlation between the ARG1 gene expression and the CD8 T cell infiltration. And we also use the red color to indicate the significant positive correlation, and the blue color to indicate the significant negative correlation. We can also check the detailed distribution between these two variables in a specific cancer type and the tools we prefer. For example, we can check it in the head neck HPV positive cancer type and the CD8 T cell infiltration estimated by the X cell, just by click here. As you can see, there are two scatter plots. The left one is the ARG1 expression with the tumor purity, and the right one is the ARG1 gene expression level with the T cell, CD8 T cell infiltration. We can see ARG1 gene expression is negatively associated with CD8 T cells, which is what we expected because ARG1 is predominantly expressed on a immunosuppressive cell type, and this negative correlation is not because ARG1 is specifically expressed on the tumor cells, as because if you look at this scatter plot, ARG1 gene expression is negatively associated with tumor purity. And this is how we use the immune gene modules on cancer research. Besides the gene expression, we could also check the relationship between mutation on a gene and immune cell infiltration. So we can go to the mutation modules here. As we all know that TP53 is a tumor suppressive gene. So we can use the TP53 as an example here. We can check the CD8 T cell infiltration between the TP53 mutant tumors and the TP53 wild-type tumors. Similar to the gene module, TIMER2 also output a similar format of the heat map, but the difference is that the value in this heat map represent the log4 change of CD8 T cell infiltration in mutant samples over the wild-type samples, and the red color indicated a significant high level of CD8 T cell infiltration in TP53 mutant tumors, and the blue color indicates that significant lower level of CD8 T cell infiltration in TP53 mutant tumors. And we also could check the detailed distribution of CD8 T cell infiltration in these two groups in a certain cancer type and a algorithm we prefer. For example, we can click here. So the TIMER2 will output this box plot to show the CD8 T cell infiltration in the wild-type group and the mutant group. And this is how we use the mutation module in cancer research. Moreover, we could also look into the association between somatic copy number alteration on a gene and immune cell infiltration. So we can use the SCNA module to do that. For example, MYC has been reported to be involved in a mechanism that can evade NK cell immune surveillance. So we can use the MYC-A as an example here and check its association with NK cell. So we can select NK cell. Here we are trying to look at the NK cell infiltration between the MYC-A amplified tumors to the MYC-A diploid tumors. Similar to previous module, TIMER2 also output this heat map, and the difference is that the value in this heat map represent the log4 change of NK cell infiltration between the MYC-A amplified tumors and the MYC-A diploid tumors. And the red color here indicates the significantly high level of NK cell infiltration in MYC-A amplified tumors. And the blue color means the significant low level of NK cell infiltration in MYC-A amplified tumors. And we could also check into the detailed distribution of NK cell infiltration in specific cancer type and the NK cell infiltration estimated by the tool we prefer. As you can see, this box plot shows the NK cell infiltration estimated by the sample sort among the different samples in the lung squamous cancer. From this figure, we can see amplification of MYC-A significantly decreased the NK cell infiltration in tumor, which is consistent with the study we just mentioned. And this is how we use the SCNA module in our research. In addition, tumor-infiltrating immune cells have been reported to affect patient overall survival. Here in this immune outcome module, we can interactively explore the association between infiltration of interested immune cell type and the patient overall survival. For example, we could explore the relationship between the CD8 T cell infiltration and the patient overall survival while controlling age, stage, gender, and the SOX10 expression, those confounding factors. And then we can submit the query. So TIMER2 also output the same heat map format. And the value in this heat map represent the coefficient of CD8 T cell infiltration in this Cox-pH regression. And the red color indicates the CD8 T cell infiltration is significantly associated with increased risk of patient. And the blue color means significant association of CD8 T cells with the decreased risk in patient. From this heat map, we can see CD8 T cells are good for patient survival in several cancer types, including the HPV-positive head and neck cancer and the melanoma cohorts. And we can also further check the relationship between the CD8 T cell infiltration with the patient overall survival in specific cancer type that we are interested in. And for example, we can just click here. As you can see, first, it will generate the detailed information on this Cox-Page regression and also output this Kaplan-Meier plot. In this plot, patients were separated into 4 groups by the SOX10 gene expression and CD8 T cell infiltration. And users can also set the cutoff by themselves. Here, for the expression and infiltration, we just use the median as the cutoff to separate the patient. And if you just change the cutoff, you can see the figure will change accordingly, as well as the p-value. So you can see from this figure, only when the SOX10 expression is high, we can see a beneficial effect of CD8 T cells on melanoma cohorts, which is consistent with the role of SOX10 because SOX10 has been reported to can promote the T cell mediated tumor killing. And this is how we utilize the immune outcome module in our research. Furthermore, there are several modules in TIMER2 for investigating the non-immune related association between tumor features in TCGA cohorts, which are in this exploration component. For example, you can check the differential gene expression between tumor and normal samples. So we can type, for example, we can use a cancer testis gene, for example, which is OTOA. You can see TIMER2 will output several box plots, and the red one are the tumor samples, and the blue one is the normal samples. So from this figure, we can see this cancer testis genes is significantly higher in tumor samples, which is consistent with the characteristic of these genes because it is a cancer testis gene. And we could also check the relationship between a gene expressions with patient overall survival. And we can use several marked genes for CD8 T cells as an example for the CD8A, CD8B, and Gram-zone. And we can control the age and the stage, those confounding factors. And you can choose more if you want. And you can see overall, the marked gene expression is good for patient overall survival. And also we can check how a mutation on a gene will affect the expression of other genes. For example, we can check TP53 mutation and test whether the marked gene expression of CD8 T cells are different in the TP53 mutant group and TP53 wild type group. You can see sometimes the TP53 mutations has a high expression of CD8 T cell marked gene, and for example, in the breast cancer. And sometimes the TP53 mutant samples have low expression of CD8 T cell marked genes, for example, in HPV positive head and neck cancers. And we could also check the expression between the interested gene to other genes. For example, we can check the BRCA1, which with the different gene, whether it's associated with CD8 T cell and CD8 marked genes. So we can input here. And you can include multiple genes if you like. You can see Like the BRCA gene expression is positively associated with marked gene expression of CD8 T cells in several cancer types. And this is the features TIMER have to explore the non-immune related association in TCGA cohorts. TIMER2 also has the third component, which is estimation component. In this component, user can upload their own expression profile, which is the TPM value. And the TIMER2 can estimate the immune cell infiltration for every samples that user uploaded. And Timer will use 6 algorithms we just described. And for example, we can download the example file here and also upload the example file to the website. And this is 5 samples from the TCGA lung cancer, and we can choose the species for these samples, which is human, and also choose cancer type, which is lung adenocarcinoma, and click Run. So after several seconds, you can see first there is a table. It contains the immune cell estimation by different algorithms. You can see timer, cybersort, and etc. And the Excel. And these are for all the samples user uploaded. And TIMER2 also output these two figures, first to compare the immune cell infiltration among different samples, and also shows compare the proportion of these different immune cell type within the same patient. And this is how you can use the estimation modules to do the immune cell estimation analysis. And this is all for the TIMER2. Thanks for your watch.